And finally, when you look at complement, remember we're talking humoral immunity. Everything is drawn to the B cells and the antibodies they produce. So our antibodies are going to trigger complement, whatever they're bound to. They trigger enough of the complement cascade. They're going to induce uptake by some cell. Here they're showing you get some complex. Whatever this is, these antibodies, this IgG is bound to, is too small to be opsonized. So whatever it is bound to, it's neutralized. But notice 1, 2, 3, 4, it could be more, and you get antibodies binding it up, forming these what they call complexes. It's going to be a large chain of antibodies and this antigens. Whether it's on a straight chain like this, bound up like that, doesn't matter. Because it's now stable, and these antibodies can act as a platform to recruit in C1q. C1q will bind to these antibodies that are a nice arrangement. It is stable. It will bring in C1R, C1S, which brings in C2, which brings in C3, and B will deposit, A will go off, activating cells. A neighboring cell, some neighboring cell will then have on its surface CR1. Remember we've talked CR1 before. CR complement receptor. Turns out C3b binds to CR1. It will trigger the uptake by some neighboring cells. What's interesting is one of the cells in our bodies that have an extremely high level of CR1s on its surface, erythrocytes, your red blood cells. So the red blood cells that are floating through your blood, delivering oxygen, also can bind up these complexes. Now keep in mind, the complex shown here is not drawn to scale to the red blood cell. If it was a true complex, the true complex would be smaller. Hundreds of these complexes could fit in this one little blue rectangle. Okay, so none of this is drawn to scale. Keep that in mind. They're just drawn this way so you can visually see it. So it'll bind to the surface. Red blood cells will bind up these complexes, which sometimes just can spontaneously happen. Sometimes, yes, they bind up stuff, not you. The red blood cells will go through the liver, the spleen, and anything on their surface will be removed by epithelial cells there, which will have FCR, their own little antibody receptors. The antibody receptors have a higher affinity for the antibodies than complement receptor has for complement, so it'll pull them off the surface. These will then be phagocytized, digested down, and the red blood cell release to flow back into the bloodstream. When you look at the different types of receptors, and no, please, for the love of God, do not try to memorize these, try to learn these. That's for grad school. Here, just know different structures. They're all based on immunoglobulin domains, and they have different binding affinities. Okay, remember, affinity means attraction to. Okay, the higher the affinity, the tighter the bind will be. Different receptors, G1s, GE, GA, GM, and they're going to be found on different cells. Now, here we see a bacteria. Bacteria has been opsonized with antibodies. The opsonized antibodies trigger complement to be laid down, C3b. So we see the beginning of the complement cascade. Well, on the surface of macrophages, amongst the many different receptors present, Fc receptor to bind fixed antibodies, and then a complement receptor to bind C3b. By binding these different receptor types, it will trigger phagocytosis. Whatever is bound will be taken up. Here it's the bacteria. Bacteria will be taken up in a lysosome, which will become a phagolysosome. Taken up in a phagosome, which will become a phagolysosome as everything here is digested, broken down, and then presented on MHC class 2, just in case. Now, why does it take bound antibodies? Why can't any free antibodies trigger all of this that we've been talking about? Well, because free antibodies that are floating in your blood will come into contact with macrophages. They will bind different receptors, FCs, and be released. Because since it's not neighboring receptors that are being bound, that will then stabilize each other, as we see here. Antibodies bound, opsonizing a bacterium, are stable. They will stably trigger neighboring Fc receptors, which will trigger a response by the macrophage. Now, sometimes, as we see here, you get a parasite. Parasites are too big. These little fluff balls you see there are actually macrophages. One macrophage, one giant, parasite larvae. Multicellular parasitic larvae, individual macrophages. Too big to be phagocytized. So, IgE coats the parasite, triggering these macrophages to basically coat the outside of the parasite, because they're going to bind, their Fc receptors are going to bind to the coated IgA, IgE, IgG that's on the surface of the parasite. And, well, since the macrophages can't phagocytize this overly large thing, instead of phagocytizing and digesting, they are going to spit out their lysosomes onto the surface of the parasite. Start digesting it outside, breaking it down, and then the macrophages can phagocytize the soup, the junk of the broken down larvae. We also see that Fc receptors are going to be on the surface of natural killer cells. So, antibodies bind to the surface of a cell. Whether it's one of your cells that's showing signals of being virally infected, or you've had a transplant. Some foreign cell is now inside of you. Antibodies will bind under normal conditions. Antibodies binding to our own cells will be removed quickly. But here they're not being removed because there's some foreign antigen holding them there. Natural killer cells will bind to these antibodies. Enough of their Fc receptors get triggered, stably bound to the surface of a target cell. It will trigger the natural killer cells to release cytokines that tells this target cell you need to die. Now notice here the cells become polarized. Here the Fc receptors on this side are being triggered, whereas the ones up here are not being bound. So that calls for a cytoskeletal rearrangement, and then all the lysosomes and granulosomes will travel to here and disgorge here, triggering apoptosis. Now, mast cells. Mast cells are what cause everybody to get runny noses when their allergies go haywire. All these little blobs you see there inside this one cell is histamine, just waiting to be dumped out. Well, Turns out mast cells have FC receptors triggered by IgE. Remember, IgE is going to be on mucosal surfaces, your sinuses, your eyes, eye sockets. Well, enough IgE gets released, it will, or won't trigger much, but if that IgE is bound to something, pollen, it will be stably linking quite a few of those FC receptors, and then the next thing you know, your sinuses are swelled up, your eyes hurt, oh my god, I can't breathe, I hate 3 pollen.